Hello class, this is our lecture on cellular respiration in plants. Let's get started. Cellular respiration is essentially the release of energy from glucose molecules that are broken down to individual carbon dioxide molecules. Overall, it is an aerobic process, meaning that it needs oxygen. Cellular respiration can essentially burn a diversity of carbohydrates, proteins, and/or fats, but we typically focus on the carbohydrate that is glucose. The overall formula is glucose plus oxygen as an input and carbon dioxide, water, and energy as an output. For an example of a plant that burns something besides sugars, this plant on the screen is called skunk cabbage, and it actually burns proteins during respiration to warm up and attract pollinators. There are three factors that can affect the respiration of a plant. The first is temperature. If it's too hot or too cold, this can make enzymes work incorrectly or inefficiently. Number two is water. Respiration rates are typically faster if there is more water in the system because water is the medium in which enzymatic reactions take place. And lastly is oxygen. We're talking about aerobic respiration here, which requires oxygen in order to generate ATP. When we learned about photosynthesis, we talked about electron carrier molecules, and now we're going to learn a couple more. Recall that electron carriers are important because they carry electrons to an electron transport chain. The electron carrier molecules that we're going to talk about for respiration includes NADH and FADH2. Our roadmap for cellular respiration is as follows. You can see that one of the reactions occurs outside of the mitochondrion and the other two occur inside the mitochondrion. All eukaryotic cells use mitochondria as a way to break down food products into ATP, which is a usable energy source. For the purposes of this lecture, we're going to be tracking the path of a single glucose molecule as it gets transformed during the course of one round of cellular respiration. And remember, this is aerobic respiration because we're talking about oxygen being present. Stage 1 is glycolysis. This occurs out in the cytoplasm, or more specifically, the cytosol that's found outside of the mitochondrion organelle. What happens? One glucose is broken down into two pyruvic acids. As a byproduct, we're also going to make a little bit of ATP, and we're going to load up a few electrons into a few electron carrier molecules. It's not a lot, but it's enough to keep the process going. During glycolysis, a glucose molecule is split into two 3-carbon pyruvate molecules. This transformation is important as we start to see the products of respiration take shape. The enzymes of the glycolysis reaction extract some of the potential energy that is stored in glucose. The process ends up yielding 2 ATP molecules and 2 electron-carrying NADH molecules. Glycolysis requires an input of 2 ATPs to activate the glucose before this process even begins. You've probably heard of the phrase, spend money to make money. So the cell is going to invest a little bit of ATP because later on in this process, we're going to get a lot more bang for our buck. Now in total, 4 ATP are produced during glycolysis, And remember that 2 were used to start, so the net yield is 2 ATPs overall. It's not much, but it's enough to keep us going into the next stage. Note that glycolysis reactions don't actually require oxygen. We haven't used oxygen yet. Therefore, glycolysis can occur in anaerobic conditions, which is conditions in which there is a shortage of oxygen. Glycolysis yields 2 ATP molecules, 2 electron carriers loaded up with electrons, and 2 pyruvates. Next up, let's see what happens to those pyruvates in the citric acid cycle. Now, the citric acid cycle is also called the Krebs cycle. And now we're going to move into the mitochondrion, specifically to the matrix. What happens? Well, the water is going to be used, and pyruvic acids are going to be broken down. And in going through energy transformations, we're going to end up forming carbon dioxide gas that leaves as a waste product. The citric acid cycle ends up producing 2 ATPs and a lot of electron carriers. The reactions of the Krebs cycle and the electron transport chain do require oxygen gas. These reactions yield much more ATP than did glycolysis. In the Krebs cycle, carbons are stripped away and bind with oxygen to yield this carbon dioxide waste product. The 2 pyruvate molecules produced during glycolysis undergo an oxidation reaction as they enter the mitochondrion. Remember, oxidation in chemistry means loss of electrons, which means we're going to release energy. A carbon atom is stripped from each pyruvate, and this leaves the cell as carbon dioxide molecule. At the same time, NAD plus is reduced to NADH. And remember, reduced means it gains electrons, and energy is therefore required to achieve this goal. Through this process, each pyruvate molecule, now with only 2 carbons, is converted to an intermediate form called acetyl CoA. CoA means coenzyme A, and this preps the pyruvate to get it ready for the actual citric acid cycle. Acetyl CoA has 2 carbons because 1 carbon from the original pyruvate left the organelle as carbon dioxide gas. Now each acetyl CoA molecule can enter the Krebs cycle, and during the course of this Krebs cycle, 2 acetyl CoA molecules are oxidized, giving us carbon dioxide gas, a little bit of ATP, and a ton of electron carriers. A ton of electrons are loaded into these molecules, which can be delivered to the next step of respiration. So far, the aerobic respiration of 1 glucose molecule has given us only 4 ATP. That's not very much, and it's not enough to keep things alive. But 10 electron carrier molecules have been produced, as well as a couple more. That's lots of electron carriers. What happens to these electron carrier molecules? Well, they deliver electrons to the electron transport chain. And it's during this part of the process that tons of ATP is produced. In stage 3, which is electron transport, this occurs on the inner membrane of the mitochondrion. What happens? Well, there's an electron transport chain that releases the energy that your cells will need to make the most of their ATP. Now, while these electrons are moving down the electron transport chain, it's actually going to drive the pumping of hydrogen ions into the intermembrane space, which forms a hydrogen ion gradient. Those ions move from high to low concentration and drive the machine that makes ATP. In fact, it makes a lot of ATP. About 34 are typically produced. During electron transport, oxygen is used and water is produced as a waste product. Let's take a look at this step by step. So NADH and FADH2 donate their electrons to the electron transport chain, where energy from the electrons is used to produce lots of ATP. As electrons travel through the transport chain, carrier molecules use the potential energy of the electrons to transport hydrogen ions into the intermembrane compartment. Sound familiar? This is the exact same process that occurred during the light reactions of photosynthesis. At the end of the transport chain, electrons are donated to an oxygen atom, which combines with hydrogens to form water as a waste product. In a sense, we can think that oxygen acts as an anchor, pulling electrons towards it in an organized way. The hydrogen ions move down their concentration gradients from the intermembrane compartment into the matrix through ATP synthase machinery, thus producing ATP. The electron transport chain produces 34 ATPs overall. That's a lot. That's enough to keep most living things functioning. When we're studying for the exam, make certain that you understand the role of these electron carrier molecules and how they ultimately lead to powering ATP formation. Cellular respiration of 1-glucose yields about 36 ATP. Glycolysis and the Krebs cycle each produce 2, and electron transport chains produce 34. However, transporting NADH into the mitochondrion requires 2, making the total production of ATP equal to about 36. And these numbers are not absolute. It changes depending on the conditions in which respiration is occurring. So there are actually additional metabolic pathways that plants can use as well. And this refers to what we call secondary metabolism. These are processes that aren't required for normal growth and development, but they can be essential to plant survival. For example, they can help plants survive and persist under difficult environmental conditions. For example, the secondary metabolites might give plants their color, aroma, or poisons. This gives them a competitive edge. Substances such as codeine, nicotine, lignin, salicin, camphor, menthol, and rubber are all examples of products made by secondary metabolism. And you don't actually need chlorophyll in order to benefit from photosynthesis. Here is an example of a fabulous parasitic plant called Sarcodes. It's also referred to by its common name of snow plant, and it grows in the Sierra Nevada mountains. It's parasitic on a fungus that lives underground, and the fungus grows as a specific mycorrhizal partner to the surrounding conifer trees. Carbon tracking shows that sugars from conifer roots actually end up in this parasitic plant. So why is it bright red? Well, it's to attract pollinators, because these flowers are pollinated by hummingbirds and bees with short tongues. One study demonstrated that Sarcodes can also self-pollinate itself successfully. a very cool example of benefiting from these sugars. That is all things cellular respiration. We've now discussed first photosynthesis and then respiration, and I hope that these important metabolic processes make sense to you. Remember that respiration is broken down into 3 steps, which is glycolysis, the citric acid cycle, and the electron transport chain. Thank you for listening. As always, please email me anytime if you have a question.